Hello students, how are you? I hope that you will be all very fine and you will be busy in your studies. Now, today we are here to give you a one-shot video or to give you an idea or overview of the most important topics with respect to IIT biology section. Okay, so today we will be mainly focusing on covering the most important topics of biology with respect to biology with respect to IIT exam. Okay, so let's see. So your biology section or the syllabus, the whole syllabus of biology, it is basically divided into two parts. We know that biology is quite a factual subject, as well as there are conceptual topics also. But yes, so we have actually divided biology into two parts. One is factual, one is conceptual. Factual topics includes the first unit of your 11th standard, that is classification unit, microbes in human welfare, morphology and anatomy of plants and animals, plant growth and development, human physiology. All these chapters, they have lots of facts in it, and you know that facts, they don't need a rigorous understanding, but they need rigorous revision. Okay, so in order to cover these all chapters, you have to just write down these chapters in your copy notebook, or you can just simply tick mark on whatever book you follow and start revising them per day, so that after these 8-9 days or 1 week, it actually starts to come in your subconscious memory. Okay, so we are not going to cover these all topics because these chapters requires only your hard work, not our hard work. But yes, we'll be covering all the conceptual topics in this today's session, because it requires your hard work as well as our hard work. Okay, because these are conceptual topics. So we will start with cell biology, followed by biomolecules, photosynthesis, respiration, genetics, molecular basis of inheritance, evolution, biotechnology, and then ecology. Okay, so let's see them one by one. Let's first start with cell biology. So cell biology, this unit includes two chapters. First chapter is the chapter which includes a basic information, which gives you a basic information about the structure of a cell, different cell organelles and their functions. And then the second chapter comes, that is cell cycle and cell division. That chapter is even more important. Let's see the first chapter. Okay, let's see the first chapter. Now, in this chapter, we have to mainly get an idea of three things. What are the different cell organelles in a cell? What are the functions of those cells, of those cell organelles? And third one is how many membranes are enclosing these cell organelles. Okay, let's see. Like we know that there are different cell organelles, starting from the most important, which is nucleus. Nucleus, it contains DNA. It is the central coordinating unit of the cell. It is just like our brain of our body. It includes structure like nucleolus, where rRNA synthesis happens. Okay, this is a double membrane system. Followed by mitochondria. We know that mitochondria is a structure which is known as powerhouse of the cell, and respiration happens in this cell organelle, and it is also a double membrane system. Chloroplast is also a double membrane system, and it is involved in photosynthesis. Ribosomes, it do not have any cell wall, and it is involved in protein synthesis. Golgi body is very essential because it is involved in different kinds of functions like packaging, secretion, sorting. Also, it's a single membrane structure. It was discovered by Count Camillo de Golgi in 1800s, some 1800s. Okay. Next cell organelle is lysosome, which is also a single membrane system, and it is involved in digesting the cell debris and digesting anything which is foreign in the cell. And sometimes when the cell gets older, it digests the whole cell also because it contains hydrolytic enzymes which are active at acidic pH. It is involved in regulated cell death, which is apoptosis, Okay, now let's move ahead. There is one structure which is known as endoplasmic reticulum. This endoplasmic reticulum, it is of two types, smooth endoplasmic reticulum and rough endoplasmic reticulum. Smooth endoplasmic reticulum is involved in lipid metabolism, lipid breakdown, lipid formation, or lipid or steroidal drug metabolism. Protein synthesis is a function of rough endoplasmic reticulum because it is rough. And how do this endoplasmic reticulum become rough? Because they have ribosomes attached on the endoplasmic reticulum, and we know that ribosome has a function of protein synthesis, and if ribosome is attached on this endoplasmic reticulum, that means that it is also going to be involved in protein synthesis. Clear? Now, vacuole is one structure which is very important because it is involved in the storage of excess water, minerals, and excess and some amount of those substances which are meant to be excreted later. Okay. Centrioles and centrosomes are involved in formation of spindle fibers, which ultimately helps in cell division. Okay. Out of all these cell organelles, nucleus, mitochondria, and chloroplast are double membrane system. Ribosome and centrioles are no membrane system, non-membrane system, and the remaining ones, which are known as endomembrane system, are single membrane system, which includes Golgi body, lysosome, endoplasmic reticulum, and vacuole. It is also known as GERL plus vacuole. Okay. Now next chapter. Next chapter actually is cell cycle and cell division, and we know that cell cycle and cell division is divided into two parts. First one is known as interphase, and the next phase is known as M phase. M phase is a phase where actual cell division happens, and interphase is a phase in which cell division do not happen, but the preparation of the cell division happens. Okay, it's just like a marriage, है ना? So the preparation for the marriage, it starts quite earlier, but the marriage happens at one night only. ठीक है? So this is analogous to the day of marriage, and this is analogous to the whole preparation that goes on before marriage. Okay. So in these stages or interphase, the cell is metabolically active, but it is not dividing, and it is divided into three phases: G1, S, G2. G stands for growth phase, S stands for synthesis phase, G2 stands for second growth phase. In G1 and G2, there is all lot of growth and all lot of protein, sugar synthesis that happens, and for cell division. But this S phase is very critical because in this phase, what happens? Replication of DNA happens here. ठीक है? Replication of DNA happens in the S phase of cell division, and that's why after S phase, the amount of DNA is doubled. So if we compare a cell in G1 phase and in S phase, the amount of chromosome will be same, but the amount of DNA in S phase will be double as that of G1 phase. Okay, clear? There is one more stage which is known as G0 phase. In this phase, the cell is confused that whether it should undergo cell division or not. That's why it is known as a cushioned stage, because it's a confusing state. Okay, now let's move ahead. The M phase or the actual cell division phase is divided into two parts. First one is known as mitosis, and the second one is known as meiosis. Mitosis is the phase of cell division, or is the type of cell division which occurs in somatic cells, while the meiosis occurs in germ cells as written over here. ठीक है? Now, mitosis occurs in order to replenish, regenerate the lost cell or the growth of our body. But meiosis, it happens only for one specific function, that is the formation of gametes. We know that gametes are haploid and germ cells are diploid. So in meiosis, the number of chromosome, it is halved. ठीक है, it becomes half of the parent cell. That's why it is known as reductional division. But in case of mitosis, the new cell has same number of chromosomes and same number of DNA, same amount of DNA as that of the parent cell. That's why it is known as equational division, because the cell number or the chromosome number remains equal in them. Clear? Let's move ahead. Now mitosis, it is divided into 4 different phases: prophase, metaphase, anaphase and telophase. By the way, these phases are known as karyokinesis. Karyokinesis, because here the nucleus divides, not cytoplasm. This is the phase where nucleus divides, not cytoplasm. So in prophase, we know that the chromosome, it is present in decondensed form earlier. That's why it starts to condensate in prophase. In metaphase, those chromosomes, they start to align at the equator, just like this. The chromosome, they start to align at the equator, okay, of the cell, and spindle fibers, they start to attach them on the kinetocores of the chromosomes, okay. Clear? And in anaphase, these spindle fibers which were formed by these centrioles, they actually split the chromosomes into 2 chromatids. So one chromosome is divided into 2 chromatids, and these 2 chromatids, they start to move on opposite pole. They start to move on opposite poles. Clear? And in the telophase stage, the nuclear membrane around these 2 sides are formed, and that's how the karyokinesis or the nuclear division ends. Now, nuclear division is not the end of cell division, because one cell can't have 2 nucleus if they are normal cells. So that's why their cytoplasm needs to be divided. And in order to divide a cytoplasm, we have to do a stage or a process known as cytokinesis. And cytokinesis, they are different in case of animals and plants, because animals don't have cell wall while plants have a cellulosic cell wall. So in animals, the moment cell has to undergo cytokinesis, the cell membrane, it starts to form a furrow. The cell membrane, it starts to form a furrow which gradually comes inside. Okay, which gradually starts to come inside and move toward the center, and hence it joins at the center, and hence the one cell containing 2 nucleuses, they divide into 2 cells containing one nucleus. Clear? This is known as cell furrow method. Now the second one is known as plants. And plants, we know that there is cell wall. That's why there is no furrow formation here. Rather, a cell plate is formed at the center of the cell, which contains calcium and magnesium pectate, and this cell plate, it starts to move from centered towards the end of the cell, and hence at one point it joins the periphery of the cell, and one cell is divided into 2 cells. Okay, clear? This thing should be clear to you. Now let's move ahead. Now, meiosis is the next thing or the next stage that we are going to discuss here. In meiosis, we know that the diploid content has to get divided into haploid content. Okay, the diploid content has to get divided into the haploid content. That's why this meiosis, it takes place in 2 phases. In one phase, the chromosome number is halved, or in meiosis 1, chromosome number is halved. While in meiosis 2, DNA content is also halved. Clear? That's why in meiosis there are 2 rounds of cell division that happens, meiosis 1 and meiosis 2. Now in meiosis 1, there are again 4 phases: prophase, metaphase, anaphase and telophase. In prophase, there are separate 5 phases known as leptotene, zygotene, pectine, diprotene and diakinesis. Leptotene is not that important phase, but from zygotene it becomes very important. In zygotene, there occurs a process or a phenomenon known as synapsis. In synapsis, the homologous chromosomes, they start to come together, they start to align one after another or at the front of each other, and this is known as synapsis, which is mediated by a protein complex known as synaptonemal complex, and hence these homologous chromosomes, they start to align near each other, or else we can say that they start to look like this. 1, 2, 3, 4 chromosomes, they start to align just in front of them. Clear? They start to align just in front of them. So these are homologous chromosomes. These are homologous chromosomes. Okay. Now, after homologous chromosomes they come together, there occurs a process known as crossing over, which leads to recombination. And it is done with the help of an enzyme known as recombinase. It is done with the help of an enzyme known as recombinase. Here there is a mutual exchange of genetic material between the homologous chromosomes, which is important for variations to happen in at gametic level. So when there is exchange of genetic material, at that time the chromosomes, the homologous chromosomes, they start to look like this. Okay, it seems that they are very close to each other and the mutual exchange of genetic material is also seen, and this is known as chiasmata. And now at the end, diakinesis, these chiasmata, they start to move away from each other, which is known as terminalization of chiasmata. Okay, this was the prophase 1 of meiosis 1, which is very essential and which is very different from that of mitosis also. Now the cell enters metaphase 1, in which homologous chromosome, they condense fully, but they are still aligned in the front of each other like this. And in anaphase 1, what happens is that these chromosomes, they are attached to the spindle fibers and they start to move on opposite poles. Now here there is a catch. In anaphase 1 of meiosis 1, homologous chromosomes are moving on opposite poles. But in the anaphase 1 of mitosis, chromatids were moving on opposite poles. This is very important difference between meiosis and mitosis, that in the anaphase of mitosis, chromosome break down into chromatids and they move to opposite poles, but in anaphase 1 of meiosis, chromosomes do not break, but rather one of the homologous pair of chromosomes, they start to move on opposite poles. Okay, and then in telophase 1, the chromosomes, they had the one chromosomes of each pair of homologous chromosomes, they have moved on the opposite poles and nuclear membrane forms on them. Okay, nuclear membrane forms on them. Okay, clear? Now chromosomes number has been halved from 46 chromosomes, let's say in humans 23 are present now in one cell. Okay, but we know that on each of the chromosome there is double the number of DNA. Okay, that's why in order to half the number of DNA, meiosis 2 happens, which is same as that of mitosis. Okay, so it occurs as prophase 2, metaphase 2, anaphase 2 and telophase 2. Clear? Now let's move on to next chapter, that is biomolecule. Biomolecule is a very important chapter because it has lots of concepts in it, but At the same time, it has lots of facts in it. Okay? Like in biomolecules chapter, there are lots of biomolecules which are polymers, but they are made up of monomers. We know that polymers are made up of monomers, but yes, we have to study these things, that which polymers are made up of which monomers, or which monomers combines to form a specific type of polymer. Okay? Then we have to see enzyme kinetics, nucleic acid structure, and tables and diagrams. This you can take as homework, because this chapter has lots of tables and diagrams in it. Diagrams you can skip also, but tables are very important, specifically the table of secondary metabolites. Okay, let's move ahead and see this chapter. Let's see. So we know that biomolecules are divided into two parts, biomacromolecule and biomicromolecule. Lipid is not placed in any of these categories, because lipid is a different kind of molecule. Its weight is very less, that's why it is a micromolecule, biomicromolecule, but it forms aggregates, and that's why it is not able to separate itself as a biomicromolecule. It comes under the fraction of biomacromolecule. But except lipids, other molecules can be grouped under biomicromolecules or macromolecules. Monomers of all the biomolecules will be placed under micromolecules, and their polymers will be macromolecules. Like if I tell you about sugars, their monomers are known as monosaccharide. They polymerize to form a polysaccharide. Okay. Now, glucose is a monomer. Fructose is a monomer. Glucose, it polymerizes to form starch. It can also form glycogen. It can also form cellulose. Okay, iodine test of starch, glycogen and cellulose, it gives blue, red and no color respectively. Why cellulose gives no color? Because cellulose do not have a helical or a branch structure. Cellulose is a straight chain molecule. Okay? That's why it's do not give any color on starch test. Clear? Clear? Let's move ahead. Fructose, polymer of fructose is known as inulin, and polymer of N-acetyl glucosamine is known as chitin, which is present in the cell wall of fungi, and it is also present on the exoskeleton of many insects. Okay. Now there is something known as disaccharide. What do you mean by disaccharide? Disaccharide means a sugar containing two monosaccharides. Okay? Like lactose is made up of glucose plus galactose. Sucrose is made up of glucose plus fructose. Maltose and trehalose are made up of glucose plus glucose. Two glucose molecule basically. Among all these disaccharides, we know that most of the sugars are reducing in nature, but some sugars whose aldehyde or ketone group is not free, those sugars are actually non-reducing sugars, and among them sucrose and this trehalose is a non-reducing sugar. Okay? This is an important point to remember. Let's move ahead. Now, amino acids. What do you mean by amino acid? Amino acids are those molecules which contains an amine group and a carboxyl group. Okay. Along with it, it has one hydrogen atom, not ion, and there is an R group. So this R group is a variable group, and we have different types of amino acids in nature based on the differences in this R group, because other groups remains same always. Okay? In nature, we have 20 types of R groups. That's why we have 20 types of amino acids. Clear? Out of these 20 amino acids, you have to mainly remember the structure of 3 of them, which are mentioned in your book also, syllabus also. Those are glycine, alanine and serine. Glycine has R group as H, alanine has R group as CS3, serine has R group as CS2OH. Other things remains the same. Clear? Because NH2, H and COH they always remain the same. Just R group is different. R group is H here in glycine. R group is CH3 here in alanine, and R group is serine here in this CH2OH. Clear? Now, this, if this R is H, then this amino acid will look like what? CH, H, COOH and NH2, COH and NH2. It has two H atoms. That's why it is non-chiral in nature. All the amino acids shows chirality and optical activity except glycine, okay? And these amino acids, they polymerize to form proteins, and proteins are of four types: primary, secondary, tertiary and quaternary. Primary, secondary, tertiary and quaternary. Okay. Now, primary structure is a straight chain, secondary structure is a little bit folding, and tertiary structure, it has 3D folding in it. But in quaternary structure, there is more than one protein chain which combines to form quaternary structures. If I try to give you an example of all these things, like primary structure, if you will heat any structure of protein, be it secondary, tertiary or quaternary, they will always come in its primary form, and in primary form we are able to get the positional information of amino acid very easily. Okay. Secondary structure, it is of two types: alpha helix and beta pleated sheet. Alpha helix and beta pleated sheets, they are stabilized by hydrogen bonds. Alpha helix example will be what? The keratin of your hair. Beta pleated sheet, the example will be fibroin of silk. Clear? And tertiary structure, all the enzymes are present in its tertiary structural form. Many enzymes are present in their secondary structural forms also, but yes, most of the enzymes are present in their tertiary structural forms. One very common example is myoglobin. Okay. The quaternary structure of protein has a very great and a very common example, that is hemoglobin. Hemoglobin is made up of 4 chains, 2 alpha and 2 beta, and I have said that quaternary structure is what? Where more than one type of polypeptide chains combine. So hemoglobin is that structure. Okay? Clear? Let's move ahead. Now, amino acid, they have a very unique feature that they have two ionizable groups. Okay. One is an acidic group and one is a basic group. We know that the basicity of base, it increases at acidic pH, and acidity of acid increases at basic pH, because acid, it actually tends to release H plus ions, and which is more facilitated in the basic pH. But in the acidic, in the acidic pH, we know that the base will show its basicity more often, because base, it tends to accept H plus ions, and it will only accept H plus ion if there are plenty of H plus ions surrounding the amino acid or in the surrounding environment. Okay. But so we can say that at low pH, the charge on this amino acid will be positive, because it will accept H plus ions to become positively charged, and it will be neutral at acidic pH. At very high pH, this will be neutral, but this will be negatively charged. So at very high pH, the amino acid as a whole will have negative charge. At very low pH, the amino acid as a whole will have positive charge. But at a certain optimal pH, there will be equal positive and negative charge. So the net charge will be equal to 0. And that stage is known as Zwitter ionic stage of amino acid. And the pH at which Zwitter ionic stage is reached is actually known as PI, or isoelectronic pH. So at isoelectronic pH, amino acid will have no net charge. That stage is known as Zwitter ionic stage. And yes, and at deuterionic stage, we know that this amine and this this amine and this carboxyl group will contain equal and opposite charges. Okay, clear? Let's move ahead. Now, enzyme activity. We know that enzymes are what? Enzymes are mostly protein. Not all enzymes are protein. Like there is an enzyme known as ribozyme, which is not a protein. It's an RNA molecule. But yes, most of the enzymes are protein in nature. And enzymes are specific in nature. ठीक है? Enzymes are very specific in nature, which means that one enzyme binds with only one type of substrate. And it binds with substrate to form enzyme substrate complex, followed by enzyme product complex, and then enzyme and product, they get separated. Clear? One property, let's see some of the properties of enzymes. One property of enzyme is that it is not degraded in this whole reaction. Second property is that enzyme do not change the equilibrium constant of any reaction, but it increases the rate of the reaction. Clear? So this is the thing. Now, enzymes, we know that enzyme reacts with substrate to form enzyme substrate complex, then product will get separated here. But we know that enzyme, initially they will be present, not enzyme, substrate, it will be present at some potential energy state, and product will also have some potential energy. Okay? If this substrate has to form product, then initially it has to activate itself, and it will get activated if and only if it absorbs certain amount of energy known as activation energy. Otherwise, it will not form product. It is not like that ki substrate, it has some potential energy and it releases some energy to form product. No, this will never happen. Substrate, even if it is at a higher energy level, then also it has to absorb at a certain amount of energy, which is known as activation energy, to form a transition state, and then from this transition stage forms a product. Okay, clear? So this is the thing. And how enzyme increases the rate of reaction? Because enzyme decreases the activation energy. Enzymes, it decreases activation energy. Enzymes decreases activation energy. Enzymes decreases activation energy, and hence it increases the rate of the reaction. Clear? Now let's see what's next. Enzyme, in this chapter or in this topic, there is a concept of enzyme inhibition. Anything which decreases the rate of enzyme catalyzed reaction are known as inhibitors. And in your syllabus, only one kind of inhibitor is mentioned, that is competitive inhibitor. Okay? Competitive inhibitor is a type of inhibitor which looks similar to substrate, and it competes with the substrate to bind on the active site of enzyme. Okay, clear? So it means that inhibitor or competitive inhibitor specifically, they have same shape as that of substrate, and they compete with substrate, normal substrate, to bind with the active site of enzyme. Clear? Clear? Now, the examples of competitive inhibitor will be inhibition of succinic dehydrogenase by malonate and oxaloacetate. This is one very important example. And there is also one example which is methanol poisoning. So ethanol and methanol are produced by alcohol dehydrogenase. Mainly ethanol is produced, but once the amount of ethanol it starts to increase, at that time alcohol dehydrogenase enzyme, it starts to produce methanol oils also, which actually competitively inhibits the activity of this enzyme. Sulfur drug is also one example of competitive inhibitor, but it is not there in your syllabus, so you don't need to remember it. You just need to remember this and this example. Specifically, this one is very important. Okay, let's move ahead. Enzyme. If we talk about the structure of enzyme, we know that enzyme, it is known as the whole enzyme, or the active enzyme is known as holoenzyme. Okay? Whose 90% of part is comprised of apoenzyme, which is a proteinaceous, which is main proteinaceous part. It has the active site. But in order to activate this apoenzyme, we have to add some cofactor in it. Okay? And that cofactor can be an organic compound, or else it can be an inorganic compound. Okay? If it is organic and tightly bound to enzyme, then it is known as prosthetic group. And the example of prosthetic group is HEM, which is present in peroxidase and catalase enzymes, which is important to break down these hydrogen peroxides into water and oxygen, because peroxides are not good for our health. Okay? If the organic compound is loosely bound to enzyme, then it is known as coenzyme. And many of the vitamins that you take from outside, they come under the category of coenzyme. So now you will be able to understand that why vitamins are important, because these vitamins, they act as micronutrients, and one of their function is to activate enzymes. Okay. If it is inorganic ion, then it can be metal ions also. So there are many metal ions which activates many type of enzymes. Such enzymes which requires metal ions to get activated are known as metalloenzymes. Like one example is zinc. Zinc activates many enzymes like carboxypeptidases, carbonic anhydrases, alcohol dehydrogenases. Okay, clear? Now, in enzymes, only one topic is remaining, that is classification of enzymes. And you know that enzymes are divided into different groups or different classes based on their functions. Like those enzymes which are involved in oxidation reduction reaction or the transfer of electrons and protons, they are grouped under oxidoreductases class. If it is involved in transfer of any functional group, then it is grouped under the class transferases. If they are involved in hydrolysis reaction or breakage of any molecule in the presence of water, then it is known as hydrolases. If they are involved in breakage of any molecule in the absence of water, then they are known as lyases. If they are involved in isomerization reaction, then they are known as isomerases. If they are involved in ligation reaction or joining of two groups, then they are known as ligases. Okay, clear? Okay, so let's see other things now in this chapter, which is nucleotide or the structure of DNA molecules. Okay, DNA is basically made up of nucleotide. What is a nucleotide? Nucleotide is made up of three structures: nitrogenous base, pentose sugar, and a phosphate group. And they are present like this. So phosphate group, you know that it is PO43 negative. Okay. This pentose sugar, it is of two types, ribose and deoxyribose, on the basis of whether the 2' position has H or OH. If it has OH, then it is ribose. If it has H, then it is deoxyribose. Let's see what is a nitrogenous base. A nitrogenous base is of two types, purine and pyrimidine. Purine has two ring structure like this. Pyrimidine has one ring structure like this. Purine are of two types, adenine and guanine. Pyrimidine is of three types, thymine, uracil and cytosine. Okay, in RNA we have uracil and in DNA we have thymine. And these all three structure, nitrogenous base, phosphate and pentose sugar, it joins to form a typical nucleotide. And this nucleotide, in this nucleotide, pentose sugar adds with nitrogenous base to form adds with nitrogenous base with the help of glycosidic bond, and this sugar, its 5' position, it adds with or it bonds with phosphate with the help of phosphoester bond to form a nucleotide. Now it can be a deoxyribonucleotide or a ribonucleotide. Ribonucleotide has all these structures except uracil, okay, and also it has OH at the 2' position, and deoxyribonucleotide, it has thymine and it has H at 2' position. Okay, and these structures, they polymerize to form DNA or RNA. Now RNA we'll see later, but DNA, its first structure or the double helical model was given by Crick and Watson, which was a secondary structure, and it was a BDNA structure. A DNA, a typical DNA, is a double helical structure which is anti-parallel, and its backbone is made up of sugar and phosphate. Sugar, phosphate, sugar, phosphate, sugar, phosphate forms its backbone. And these two backbones are joined by hydrogen bond, where A forms 2 hydrogen bonds with T and G forms 3 hydrogen bonds with C. Okay. Now, the sugar phosphate, they are joined by phosphodiester bonds. Okay. So each chain has phosphodiester bonds to join them, and both chains are joined by hydrogen bonds. Take next. So these were some of the typical structure, typical information about DNA, but specific information about BDNA is present over here in this diagram. So one 360-degree turn, the length of the DNA covered by 360-degree turn, or covered after 360-degree turn of a DNA, is known as pitch or one pitch. Okay, and one pitch contains a length of 34 Armstrong, and one pitch contains 10 base pair, and in one pitch there is a 360-degree rotation. There is a 360-degree rotation. Okay, so if there is a 360-degree rotation in one pitch, then the rotation between two base pair will be 360 by 10, that is 36 degree. And if one pitch has 34 Armstrong and one pitch has 10 base pair, then the length or the size between two base pair is 360 or is 34 by 10, that is 3.4 Armstrong. Okay, the diameter of this helix is 20 Armstrong. Okay. Now the next chapter is photosynthesis. In photosynthesis, we know that plants, they capture sunlight to produce C6H12O6, which is glucose. This is a reaction, and this reaction, it actually can be broken down into two parts. First one is breakage of water to oxygen, and second one is CO2, reduction of CO2 to C6H12O6. The CO2 to C6H12O6 is known as dark reaction. The H2O to O2 is known as light reaction, and light reaction occurs first, which is followed by dark reaction, because in light reaction there are some energy rich molecules which are produced, which are used in dark reaction to produce glucose. Okay, let's see. Now, plant, they have green parts, mainly leaf, and in the mesophyll of leaf are present chloroplast, and in these chloroplast we have stroma. The cytoplasm of your chloroplast is stroma, and these stack-like structure known as grana, and these stack-like structure known as grana. Each coin-like structure of these grana is known as a thylakoid, and these thylakoid, they are divided into two parts, the central lumen and the membrane present over here. This membrane has different proteinaceous structure known as photosystems. These photosystems are composed of light harvesting complexes. These light harvesting complexes, they are of two parts, light harvesting proteins and light harvesting pigments and light harvesting proteins. These pigment molecules can be of many types: chlorophyll A, B, C, D, xanthophyll, carotene, all those things. But the main pigment is chlorophyll A, which is a reaction center. Okay. Now, these photosystems, they can be divided into two parts based on the light or the wavelength of light which they absorb. If they are absorbing 680 nanometer of light, then they are P680. They are also known as PS2. If they absorb 700 nanometer of light, then they are known as PS1. Okay, let's see. Let's see now light reaction. Light reaction occurs in the thylakoid membrane. Okay, and they have photosystem like these. This is PS2 photosystem 2. This is PS1 photosystem 1. Now both of them have chlorophyll A molecule, and these chlorophyll A molecules, they have electrons in them which gets excited by absorbing light of 680 and 700 nanometer respectively. And these electrons, once they are excited, they are transferred to different electron carriers. Okay, they are transferred to different electron carriers, but at the end it is accepted by NADP plus, which accepts one electron and one proton to form NADPH with the help of an enzyme known as FNR or pyridoxine NADP reductase. Okay, so we are able to now produce what? We are able to now produce one of the two energy rich molecules. Okay, and we know that it's looking like a zigzag, that's why it is a Z scheme, and it's a non-cyclic photophosphorylation. And this cycle also has one thing which is known as water splitting complex activated by manganese ions, which are involved in breakage of water into 2H plus 2 electrons and oxygen. Why we need this system? Because once the chlorophyll A, once the electron of this chlorophyll A is excited, then this chlorophyll A becomes electron deficient. So that's why it needs a constant supply of electrons, which is supplied by this electron produced by the breakage of water molecules. Okay, clear? Now let's begin the next topic, which is cyclic photophosphorylation. So let's say we don't have PS2, or we don't have light of 680 nanometer, or else we don't have water splitting complex, or we don't have FNR molecule. Then we know that only PS1 is remaining, and at that time cyclic photophosphorylation happens, in which 700 nanometer of light excites electron from PS1, from the chlorophyll A of PS1, which is cycled back again and again in this system. Okay, so this is a way in which PS1 happens or cyclic photophosphorylation happens. Okay, I mean to say, this is the manner in which PS1 acts alone in cyclic photophosphorylation. Okay, now you must be wondering that what are the products that we got here? We got nothing here. You showed us nothing. Here at least you showed NADPH, but here you showed nothing. है ना? So I will tell that in both the cycles, non-cyclic and cyclic photophosphorylations, in both of them ATP is produced. How ATP is produced? Let's see. ATP is actually produced by actually breaking the proton gradient, which is created by the transfer of electrons. The electrons are transferred in different ecosystems, in different photosystems, and due to those transfers, electrons are continuously transferred from stroma to the lumen of thylakoid. Okay, due to which the H plus concentration rises in the lumen of thylakoid, which creates a proton gradient. Here it has high amount of proton, and outside in the stroma we have less amount of protons, and this proton gradient actually also creates a concentration gradient or an ionic gradient, है ना? And it actually wants to break this gradient, and it is broken down by this complex known as F0 F1 complex. This F1 is a channel protein, and this F0 is a channel protein, and this F1 is an ATP synthase. So H plus, they start to move from this system, from this F0 F1 system, and activate this ATP synthase activity of F1, which actually starts to produce ATP utilizing ADP and phosphates present in the stroma side of your thylakoid. Clear? Clear? Okay, so this is the way, or this is a manner in which ATP are synthesized in both cyclic and non-cyclic photophosphorylation. Okay, now if we compare non-cyclic and cyclic photophosphorylation, we can say that non-cyclic photophosphorylation, it happens in grana lamellae, while cyclic photophosphorylation, it can occur in grana as well as stroma lamella. Non-cyclic photophosphorylation, it has PS2, PS1, but cyclic has PS1 only. ATP and NADPH both are produced in non-cyclic, but there only ATP is produced. Here oxygen evolving complex is present, there oxygen evolving complex is absent. Here we have light of 680 and 700 nanometer both, but here only 700 nanometer light is required. Okay, now let's move ahead. Now we have produced ATP as well as NADPH, which are transferred to the stroma of the chloroplast, because in the stroma of chloroplast is going to happen what? Dark reaction. Dark reaction occurs in the stroma of chloroplast, in which ATP and NADPH are used and glucose is produced. So this is a cycle known as C3 cycle, because the first product is a 3-carbon product known as phosphoglyceric acid. It is also known as a Calvin cycle on the name of the discoverer of this cycle known as Melvin Calvin. Here a substrate is present from the beginning, which is known as RUBP, ribulose bisphosphate, which accepts one CO2. So this is a 5-carbon molecule. This is a 1-carbon molecule which is CO2. 5 plus 1 is 6. So this 6-carbon, it actually breaks down into 2 3-carbon molecules, which is phosphoglyceric acid. This step is known as carboxylation. In the next step, which is known as reduction, triose phosphate is formed and 1 carbon is released from these 2 phosphoglyceric acid. Okay, so these 2 phosphoglyceric acid, they are both 3-carbon molecule. So total 6, but 1 carbon is removed. That's why now only 5 carbon remains. Okay, so here 1 carbon is released after every cycle and 5 carbons are again regenerated. Okay, so this cycle comprises of 3 steps: carboxylation, reduction and regeneration. And in every cycle 1 carbon is fixed or released. Okay, we know that glucose contains C6H12O6, okay, which contains 6 carbon. One cycle has 1 carbon fixed. So how many cycles do we need to produce 1 glucose molecule? We need 6 cycles. Okay, so we need 6 cycles to produce 1 glucose molecule. In one cycle, in the reduction step, we use 2 ATP and 2 NADPH. In the reduction step. In regeneration step, we need 1 ATP. So total in one cycle, we need 3 ATP and 2 NADPH. Okay, and 2 NADPH. Okay, but we actually have to do 6 cycles to produce 1 glucose molecule. Okay, so in 6 cycle, 18 ATP, 12 NADPH are used to produce 1 glucose molecule in case of C3 cycle. In case of C3 cycle. Okay, now next thing. This step is the first step of your C3 cycle, which is combination of 5-carbon molecule RUBP, ribulose bisphosphate, and CO2 to produce 2 3-carbon molecule, that is phosphoglyceric acid. This is an enzyme catalyzed reaction which needs an enzyme known as Rubisco. Rubisco is an enzyme which is present, which is the most abundant protein of this whole biosphere. This Rubisco, it has dual characteristic. It can combine this RUBP with CO2 and oxygen also, okay, based on the concentration of both of them, based on the partial pressure of both of them. Okay, if CO2 is present in large amount, then it will combine with CO2 and will produce phosphoglyceric acid, 2 molecules of phosphoglyceric acid. If O2 is present in larger amount or very high amount, then it will combine with O2 to produce 1 phosphoglyceric acid and 1 phosphoglycolate. And this phosphoglycolate enters a cycle or a chain of reaction known as photorespiration, and it's a wasteful process which do not produces any glucose molecule, rather it uses ATP and NADPH, which were high energy molecules. Okay, so which is a bad thing of C3 cycle. So that's why there are some plants which have evolved a novel strategy to inhibit this reaction from happening. Okay, and that thing is known as hatch and slack pathway. Some plants have evolved hatch and slack pathway or C4 pathway in which no photorespiration happens. Okay, but this C4 cycle, it happens in two cells, mesophyll cells and bundle sheath cells both. And such plants known as C4 plants, they shows Kranz anatomy. Kranz anatomy means wreath. Okay, Kranz means wreath, which means that there is a similar pattern of arrangement of cells in them. Okay, how does this cycle look like? It looks like this. It has two cells, one is mesophyll cell, one is bundle sheath cell. In mesophyll cell, the carbon dioxide from the outer environment, it will come and it will get converted into bicarbonate ion, which will be accepted by phosphoenol pyruvate using enzyme pepcase to produce oxaloacetic acid. So the first product of this hatch and slack pathway is a 4-carbon product because it is formed by 3-carbon phosphoenol pyruvate plus 1 bicarbonate ion. So 1 plus 3, 4. Okay, that's why it is known as C4 cycle. Now this oxaloacetic acid or oxaloacetate will enter the bundle sheath cell where it will release this CO2, and hence several cycles like this will ultimately increase the amount of CO2 present in the bundle sheath cells. Okay, and due to increased CO2 count, there will be no process known as photorespiration that will happen here. Okay, because Rubisco has both the properties, but here CO2 is present in very large amount. That's why there will be no photorespiration. Okay, and here C3 cycle will happen because here we have Rubisco enzyme present. Okay, the remaining thing after it oxaloacetic acid decarboxylate here, the remaining thing which is there is pyruvate. This pyruvate will again regenerate into phosphoenol pyruvate, and then this cycle will again be continued again and again. Okay, now let's compare C3 cycle with C4 cycle. A C3 cycle, in C3 cycle the first product is phosphoglyceric acid, but in C4 cycle it is oxaloacetic acid. In C3 cycle there is only one cell involved, which is mesophyll cell, but in C4 cycle there are two cells involved, which are mesophyll cell and bundle sheath cells. Now in C3 cycle, for one C3 cycle to happen, we need 3 ATP and 2 NADPH, but for one C4 cycle to happen, we need 5 ATP and 2 NADPH. Okay. In C3 cycle, Rubisco is present in mesophyll cells, but in C4 cycle, Rubisco is present in bundle sheath cells. Okay, so with this, we are ending up the chapter known as photosynthesis. Now, we'll move on to our next chapter, that is respiration. Okay. Okay, so let's start our new chapter, that is respiration, because respiration is also the follow-on chapter as compared to this thing, photosynthesis. In photosynthesis, we have made glucose, and in respiration, we'll break down glucose in the presence of oxygen to release energy. What? To release energy. Okay. So here C6H12O6 is broken down in the presence of oxygen to release CO2 and H2O to produce energy in the form of ATP. Okay, let's see. So glycolysis, Krebs cycle and electron transport chain. These are the main 3 steps which are involved in respiration. Okay, let's see. So glucose breakdown, it occurs in a sequential manner. First it is broken down to pyruvate via glycolysis in the cytoplasm of the cell. Then this pyruvate is transferred to the mitochondrial matrix where it undergoes Krebs cycle, and then electron transport chain happens in the inner mitochondrial membrane, in the inner mitochondrial membrane, to produce ATP. Let's see. So glycolysis, it's a reaction in which glucose is broken down. One glucose is broken down into 2 molecules of pyruvic acid. So it occurs via 10 sequential steps. Glucose is broken down into glucose 6-phosphate in the presence of hexokinase, followed by its conversion to fructose 6-phosphate, fructose 1,6-bisphosphate, then glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, followed by formation of 1,3-bisphosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenol pyruvate, and then pyruvic acid. This is typical glycolytic cycle. But what is important is to note that how many ATP and NADPH are produced in this reaction. So total 4 ATP are produced, out of which 2 ATPs are used in glycolysis. So net production is 2 ATP and 2 NADH2 are also produced in this reaction. Okay, clear? So one glucose is broken down to 2 pyruvic acid producing 2 ATP and 2 NADH2. Now this pyruvic acid, if it do not get oxygen, then it undergoes anaerobic respiration, which is known as fermentation. If it gets oxygen, then it undergoes a cycle known as aerobic respiration. Okay? First, let's study if it does not get oxygen. If it does not get oxygen, then it can undergo lactic acid fermentation or alcoholic fermentation. In lactic acid fermentation, the pyruvate is converted into lactic acid with the help of an enzyme known as lactate dehydrogenase. Okay? And in alcoholic fermentation, the pyruvate is first converted to acetaldehyde with the release of one CO2 molecule. Then this acetaldehyde is further reduced to produce ethanol, okay, which is used as a beverage in some countries. In many countries, not some countries, okay. And this conversion is done with the help of an enzyme known as alcohol dehydrogenase, whose activity is increased or who is activated by zinc dipositive ions. Okay, clear? Now, so this was the case when it do not get oxygen, but when it gets oxygen, when oxygen is present, then what happens is that this pyruvate is transferred to mitochondrial matrix, where it first undergoes oxidative decarboxylation, in which pyruvate is broken down into acetyl-CoA. This is a 3-carbon molecule, this is a 2-carbon molecule, which means that 1 carbon dioxide is released in this step. With the help of an enzyme known as pyruvate dehydrogenase, which is a metalloenzyme activated by magnesium dipositive ions, and the net ATP or NADH production is written over here, which is 2 NADH2. Other than that, no ATP, no FADH is produced in this reaction. So what is the fate of this oxidative decarboxylation? It produces 1 acetyl-CoA from 1 pyruvate. But we are more concerned about 1 glucose. 1 glucose produces 2 pyruvates. So here 2 acetyl-CoA are produced and 2 NADH2 are produced here. Okay, with the release of 2 CO2. Okay. Now let's move ahead. This acetyl-CoA, it now undergoes the cycle known as citric acid cycle or tricarboxylic acid cycle or Krebs cycle, which also occurs in mitochondrial matrix, in which this oxaloacetic acid, it accepts acetyl-CoA to form 6-carbon molecule known as citric acid cycle, which further undergoes 2 rounds of decarboxylation to produce alpha-ketoglutarate acid and succinic acid. Here it undergoes decarboxylation, means release of CO2, and then this succinic acid is converted to fumaric acid, malic acid and oxaloacetic acid, which are all 4-carbon molecule, This is also 4-carbon molecule, this is a 5-carbon, this is a 6-carbon molecule. Okay. Now one very important thing that I must tell you here is that this succinic acid to fumaric acid conversion, it occurs with the help of an enzyme known as succinic dehydrogenase. This succinic dehydrogenase, it is the only enzyme of Krebs cycle which is not present in mitochondrial matrix. It is present in inner mitochondrial membrane, where it is present in inner mitochondrial membrane. And this succinic dehydrogenase, it is involved in both the reactions in Krebs cycle as well as electron transport system. Okay, clear? So in these sequence of reactions, the net release of ATP, NADH and FADH by breakdown of 1 acetyl-CoA is that it produces 3 NADH2, 1 FADH and 1 ATP by the breakdown of 1 acetyl-CoA. But we know that from 1 glucose we produce 2 acetyl-CoA. So 2 acetyl-CoA will be produced and broken down. That's why we have to multiply it by 2. Clear? Clear? So total 6 NADH, 2 FADH2 and 2 ATP will be produced by this cycle. Okay, let's see the respiratory chart. In glycolysis, oxidative decarboxylation, Krebs cycle, let's see how many ATP, NADPH and NADH and FADH are produced. In glycolysis, 2 ATP are produced and 2 NADH are produced. Check. Oxidative decarboxylation, only 2 NADH are produced. In Krebs cycle, 2 ATP, 6 NADH and 2 FADH are produced. Okay. Now, one thing that I must tell you here is that 1 NADH is equal to 3 ATP and 1 FADH is equal to 2 ATP. So if we calculate all the reaction substrates and we multiply with respective amount of ATP equivalents, then it will come out to be 38 ATP. So in aerobic respiration, total 38 ATP are produced by breakdown of 1 glucose. In anaerobic respiration, total only 2 ATP are produced by incomplete breakdown of 1 glucose molecule. Okay. The direct ATPs which are produced are known as substrate level phosphorylation, and the NADH which are produced, which are ATP equivalents, those are known as oxidative phosphorylation. Okay, NADH and FADH, they are not ATP, but they are ATP equivalents. So 1 NADH is equal to 3 ATP and 1 FADH is equal to 2 ATP. That we know. They are converted into ATP by the process known as oxidative phosphorylation, which is electron transport system, which occurs in the inner membrane of mitochondria, where NADH and FADH are broken down into NAD, FAD, NAD and FAD plus, releasing protons and 2 electrons, and these electrons again enter and move via different electron carriers to produce a proton gradient, and breakage of this proton gradient produces ATP, just as we saw in the light reaction of photosynthesis. Okay. Now, there are 5 total complexes which are involved in respiration. These 5 complexes are named as NADH dehydrogenase, FADH dehydrogenase, which is also known as succinic dehydrogenase, that was also involved in Krebs cycle. Cytochrome BC1 complex, which is known as cytochrome reductase, cytochrome AA3 complex, which is also known as cytochrome oxidase, F0, F1 complex, okay, which is ATP synthase also. Now, the electrons which are moving here in this system, they are finally accepted by what? By oxygen to produce water molecules. So the final electron acceptor in case of respiration is what? Is oxygen, which accept electron to produce water. But in case of photosynthesis, it was NADP, which produces NADPH. Okay, so likewise, the energy stored in NADH and FADH are converted into ATP equivalents. 2 ATP from FADH and 3 ATP from FADH. Okay, let's move ahead and see some other things. Like one topic in this chapter is respiratory quotient. What is respiratory quotient? Respiratory quotient is basically, it's a ratio which actually tells us that on complete breakdown of any molecule, how many CO2 is released and how many O2 are used. Okay? Complete breakdown of any biomolecule releases how many CO2 upon how many oxygen are used there. Okay? That is respiratory quotient. And knowing respiratory quotient, we can get an idea of how easily or how tough is the breakdown of that molecule. Like glucose, it has respiratory quotient of 1. Protein 0.9, fats 0.7. The lower is the respiratory quotient, the more difficult is it to break down them or to break them. Okay, clear? Now, you know that we studied this cycle or this whole respiration with respect to breakdown of glucose. But in the absence of glucose, amino acids, fatty acid, glycerol, they also can enter into this respiratory pathways at the step of pyruvic acid synthesis, acetyl-CoA synthesis, and dihydroxyacetone phosphate synthesis respectively. Okay. One more thing is that our respiration, it seems that it's a catabolic reaction, but during the course of respiration there are many intermediates which are produced also. That's why it is not strictly a catabolic reaction, it's also an anabolic reaction. So it is anabolic plus catabolic reaction, which is also known as amphibolic pathway. Okay, which is known as amphibolic reaction or amphibolic pathway. Now, the next chapter, which is very important chapter, is genetics. Genetics is something from which questions are asked every year. So that's why we have kept here this chapter and we'll study it. Okay. Now, genetics, it includes Mendel's laws, which is Mendel's law of dominance, law of segregation, law of independent assortment. Law of dominance says that if we take a pure line of two traits of one characteristic, one is dominant, one is recessive, then in the F1 only one of the trait will be expressed and that trait will be dominant. Okay. Law of segregation, what does it say? It says that during the gamete formation, the alleles of the two traits, they segregate from each other and they combine again during the formation of zygote or a new progeny. Okay? So even if one of the trait is not expressing, but still the alleles of those traits are present in the genotype of those individuals. Okay? Law of independent assortment says that if you are studying two characters at the same time, then inheritance of one of the characteristic will be independent from that of the inheritance of the other characteristic. Okay, so that is the law of independent assortment. Law of dominance has an exception, that is incomplete dominance and codominance. Segregation generally do not have any exception, but law of independent assortment, it has exception, which is known as linkage. So I have made here a typical Mendel's cross for one characteristic. It's a monohybrid cross. Let's say a pure line of capital T tall and a pure line of small t dwarf. It is mated to produce a heterozygous capital T small t, which is the F1 generation, where all the individuals are tall. Okay? Now if they are selfed, so capital T small t, if they are selfed, on selfing there is a possibility of production of 4 offsprings, out of which 3 will be tall and 1 will be dwarf. So 3:1 will be the ratio of the physical characteristics or phenotypes, and genotypes will look like 1:2:1, where 1 will be capital T, 2 will be capital T small t, and 1 will be small t. So the monohybrid cross in the F2 generation, it shows a phenotypic ratio of 3:1 and a genotypic ratio of 1:2:1. Clear? Now let's move ahead. Law of independent assortment is actually meant to study more than one characteristic at a time. Like if we take a dihybrid cross, then let's take two characteristics at the same time: height and color, height of the plant and the color of the flower of the plant. Okay? Color of the flower of the plant and the height of the plant. And take a pure line of both the dominant and both the recessive characteristic. Like both the dominant characteristics are tall and purple. Both recessive characteristics are dwarf and white. Okay. Just made them, and in the F1 progeny you will be able to see that for both the characteristic, dominant traits are expressed. Like in the F1 generation, all the plants will be tall and all the colors of the flower will be purple. Both dominant. Tall is dominant, purple is dominant. But when you will self them, or on self crossing, the F2 hybrids that will be generated, there will be total 16 possibilities of F2 hybrids, which will phenotypically segregate in these manners: 9:3:3:1, and genotypically they will be segregated in this manner: 1:2:1, 2:4:2, 1:2:1. Okay. This 9:3:3:1 is very important because 9 will show both dominant traits, 3 will show one dominant, one recessive, 3 will show one recessive, one dominant, and 1 will show both recessive. Okay, this is a thing. Like this, we have mentioned it over here. Now, if you are studying this dihybrid cross, and in the F2 generation, your ratio, you were expecting that the ratio will come out to be 9:3:3:1, but it did not came. It deviated from this ratio. Then what can you predict? You can predict that the genes are linked. Genes for both the characters are not present on the different chromosome, they are present on the same chromosome. They are linked. So for independent assortment to happen, both the characters, the genes of both the characters should be present on different chromosomes. But if they are linked, which means that the genes for the characters are present on the same chromosomes, then they are linked genes. And in case of linkage, your F2 ratio will always deviate from these two ratios. Okay, but one thing that I must tell you here is that if the genes are linked, then the number of parental combination will be more as compared to the recombinants. So recombinants will be less, parentals will be more. But the extent of the parental generations produced will vary. It will vary as per the distance of the genes on the chromosome. If the distance of both the characters, of both the genes for both the characters are very far, or if is very much, then we know that amount of linkage will be less and the amount of recombinants that will be produced will be higher in amount. And if the genes for both the characters are present on the same chromosome and they are very close to each other, then we'll show that the extent of linkage will be higher and the amount of recombinants will be very less. Okay? So if genes are close, then linkage is more and recombinant is very less. If genes are far away, then linkage is less and recombinant frequency is very high. Okay. Now, next in this chapter, there is a topic known as sex determination, and in sex determination, we mainly study what? We mainly study that whether the progeny which is going to come is male or a female. In that case, we have male heterogametry and female heterogametry. Male heterogametry means the males which are produced contains two different allosomes or two different sex chromosomes. Like in humans, we have XY. Males are XY, females are XX. Females are homogametes, males are heterogametes. Like there are insects which shows XX and X0 inheritance. Those are also the examples of male heterogametry. But there are cases like in chickens, in some birds, they show female heterogametry in which females have two different chromosomes, not males. So females have ZW chromosome and males have ZZ chromosomes. Okay. Now, there is a very important case of sex determination which is known as haplodiploid sex determination, in which males are haploid and females are diploid. In honeybee, males have 16 chromosomes and females have 32 chromosomes. Males are developed by parthenogenesis of the egg and females are developed by the trivial type of fertilization and then zygote development processes. Okay? So females are developed by fertilization and males are developed by parthenogenesis. And in this case, the males actually they don't have father and they can't have sons also. Okay? But females, they have father and son. Males can have father, males can have grandfather and grandson, but not father and son. Okay? But let me tell you one thing, that though it is haploid, but still it is fertile. In females only queen bee is fertile. This worker is not at all fertile. Okay? Now, there is one more very important topic in this chapter. Actually, all the topics are important in this chapter. One more important topic here is this one, pedigree analysis. So in pedigree analysis is basically the study of family chart or the study of inheritance of any character in the family. Okay. So let's say in a pedigree analysis, we can't write names and whether each individual is male or female. And that's why in pedigree analysis, we have assigned a particular shape and condition for particular gender and all those things, and we study it in the same pattern for different families. Okay, like males are denoted as square, female are denoted as circles. If they are mating or if they are getting married, then they are joined by a line, horizontal line. If they are having kids, then kids are denoted below them like this. If they have one male and one female kid, or one son and one daughter, then it is written over here. It's mentioned like this. Okay? If any individual is affected by any disease that we are studying, then they are shaded. Okay? If male is affected, then male is shaded. If female is affected, then female is shaded. Affected, male and female. Okay? Clear? And let me try to give you one example here. Like this. If this is a pedigree chart and this male is affected, this female is affected, this male is affected. This is a simple case of X-linked recessive inheritance because it is showing crisscross inheritance, male to female to male. Male to female to male pattern of inheritance is seen in case of X-linked recessive inheritance. Okay? Like hemophilia. Hemophilia is a disorder which is inherited like this because that is an X-linked recessive disorder. There are multiple cases of pedigree analysis, but this is one very trivial case. Okay, clear? Now let's move on to the next topic, which is disorders. Genetic disorders are of two types, Mendelian disorder and chromosomal disorders. Mendelian disorders are caused by the mutation in the gene, in any gene. Okay? And chromosomal disorders are caused because of presence of extra chromosome or because of absence of any chromosome in the whole genomic set or the whole diploid set of chromosomes. Okay. Now chromosomal disorders, they are of three types. Not three types, they are of many types, but you have to study only three types of chromosomal disorder, that is Down syndrome, Klinefelter syndrome and Turner syndrome. Down syndrome is caused due to trisomy of 21st chromosome. Klinefelter syndrome is caused due to trisomy of X chromosome. Turner syndrome is caused due to monosomy of X chromosome. What do you mean by trisomy and monosomy? Monosomy means one less chromosome. Like we have 46 chromosome. If one X is missing, if we have only 45 chromosome due to absence of one X chromosome, that is known as monosomy of X chromosome. Trisomy means one extra chromosome. Okay? Like if in a Klinefelter syndrome there is trisomy of X chromosome, what does it mean? It means that one X is extra. That individual will have 47 chromosome, 44 autosomes and XY and 1X more. 44 plus XXY is Klinefelter syndrome. Okay. How one chromosome becomes more or one chromosome becomes less in these cases? Because here law of segregation is broken down. Here there is non-disjunction of chromosomes during anaphase 1. Okay, clear? Now let's move on to Mendelian disorders. Let's see. Mendelian disorder, you have to study three Mendelian disorder. First one is known as hemophilia, second is sickle cell anemia, and third one is phenylketonuria. Hemophilia is an X-linked recessive disorder. Sickle cell anemia is autosomal recessive disorder caused by a point mutation in the globin chain, in the gene coding for globin chain of our RBC, red blood cell. Okay, actually the point mutation, it is actually in the gene. At some position we have GAG, which codes for glutamic acid, but it changes to GUG. So A changes to U. GAG codes for glutamic acid, but GUG codes for valine. So there is a change of one amino acid from glutamic acid to valine, which actually changes the shape of RBC drastically, and the shape of RBC becomes sickle shaped instead of biconcave disc shaped. Okay, now next is phenylketonuria, which is also an autosomal recessive disorder. It's a metabolic disorder in which phenylalanine is not able to get converted to tyrosine. Rather, it gets converted to phenylpyruvic acid, which starts to precipitate in our major organs like brain, heart, kidneys, and it causes damage in those organs. Okay, clear? So knowing all this information, I hope that you will be able to solve questions from these different disorders. So they will not give you questions regarding this that I am teaching you over here. They will give you a question, and then they will try to ask some numerical type of problem to you. Okay, these are the theoretical information that I have given you. You need to know these things to solve those questions, but you have to apply your brain to solve those questions, knowing these facts. Okay, let's move ahead. Now, the next chapter that we have to study is molecular basis of inheritance, in which we'll mainly study about how characters are actually expressed at a genetic or at a molecular level. Let's see. So a human or any individual has a cell, and in the cell we have a structure known as nucleus. Nucleus has chromosomes in them. Chromosomes are nothing but they are proteins and DNA. DNA wrapped in protein is known as chromosome. DNA, if we try to study DNA has many base pairs in them. Okay? Most of them are non-coding, but some of them are coding. And coding region of DNA, they are able to express themselves into mRNA, and then they are able to translate themselves into proteins. And these proteins, they start to produce enzymes, and those enzymes are involved in various reactions of our bodies, and those reactions lead to expression of different types of traits in our body. Okay, so it is basically the expression of DNA to proteins which actually leads to what? Which actually leads to expression of different types of traits in our body. Okay, clear? Let's move ahead. Let's move ahead and try to see the central dogma of molecular biology. So the first phase of molecular biology is replication. We know that in the S phase of cell cycle, our DNA, it replicates. One DNA converts into two DNA because it has to divide into two cells. One cell divides into two cells, it needs same amount of DNA. That's why we have to double the DNA to segregate properly to separate or to distribute same amount of DNA in both the cells which are going to be formed after cell division. Okay, so the DNA here, it unwinds like this with the help of an enzyme known as helicase or unwindase, and then primers are added over here with the help of an enzyme known as primase, and then DNA polymerase comes and it polymerizes both the strands. Okay, so this is the main process how our DNA replicates. But DNA polymerase, it is able to move towards the unwinding site or the side at where the things are getting unwounded. But this strand is formed opposite to the strand, opposite to the direction of unwinding, opposite to the unwinding strand. That's why this strand is a lagging strand, this is a leading strand. Why? Because here the DNA is synthesized continuously and here the DNA is synthesized discontinuously in the form of fragments. And these fragments are known as Okazaki fragments. These Okazaki fragments, they are produced in fragments, but they are joined by the help of an enzyme known as DNA ligase, okay, and that's how the whole DNA gets replicated from one DNA to two DNA we get. Okay, so this is DNA replication. Now, up to DNA replication, we are not trying to express DNA. But once it's time for the expression of DNA, then we have to convert this DNA into mRNA and then this mRNA to protein, because protein is the thing which actually governs most of the processes of our body, like the structural and functional roles of our body are mainly governed by proteins. So we have to produce proteins to express any characteristics that we have got from our parents, let's say. Okay? So first of all, DNA should be converted to mRNA. It will be done with the help of transcription. mRNA to protein is done with the help of translation. First, let's study transcription. Okay, because without transcription, we can't do translation. Transcription will be done on which site of DNA? The coding site, because the coding DNA is only the one which is going to produce protein, not the non-coding part. So coding part of DNA is known as what? Is known as cistron. Coding part of DNA is known as cistron. Cistron also have coding and non-coding regions. Coding regions are known as exon and non-coding regions are known as introns. Okay. Now, this cistron, a typical cistron, it looks like this. It has three regions. The initial region known as promoter, the middle region known as structural gene, and the last portion is known as a terminator. The strand which is 3' to 5' is known as template strand, whose information is studied by RNA polymerase to synthesize an mRNA. The other strand, the next strand is known as 5' to 3' polarity strand, is known as coding strand, who is not template, but its sequence, if you start to see see the sequence of this coding strand, then you will get to know that it has the same sequence as that of the mRNA. Because why? Because both of them, coding strand and the mRNA, are complementary to template. So both of them are complementary. It means that both of them are same in terms of their base sequences present in them. Okay, clear? So that's why we try to study all the things regarding mRNA or regarding the transcription with respect to coding strand, not the template strand, though the main transcription happens by reading the information on template strand. So RNA polymerase, it attaches on the promoter site after binding to a factor known as sigma factor. Okay, known as sigma factor. Then it starts to move like this here and start to synthesize RNA. And after synthesizing RNA, it reaches the terminator region where it binds to the Rho factor. And after binding to the Rho factor, the RNA polymerase complex, it breaks down and it releases the nascent RNA as well as itself also. RNA polymerase and the nascent RNA, it get detached from this cistron. Okay, so that's how mRNA is produced. Clear? Now let's move ahead. So in prokaryotes, we have only coding regions. That's why prokaryotes, they don't need any post-transcriptional event for the maturation of nascent RNA. Okay, but in eukaryotes, we know that cistrons of eukaryotes, they have a split gene arrangement. They have a split gene arrangement, which means that they have coding and non-coding sequences both. So we have synthesized the whole coding plus non-coding RNA, but we have to remove this non-coding RNA from the nascent RNA or the heterogeneous nuclear RNA that we have produced. Okay, now it is done with the help of a post-transcriptional modification step, which involves three steps known as capping, tailing, and splicing. In capping, a 5' cap is added. In capping, a 5' cap is added, and that 5' cap is known as 7-methyl guanosine triphosphate. In tailing, 200 to 300 adenine containing nucleotides are added at the 3' end, known as poly A tail. And in splicing, the non-coding sequences or the introns are removed from the nascent RNA known as heterogeneous nuclear RNA, with the help of a complex known as spliceosomes. And hence, after these three steps, the RNA gets matured. This HNRNA matures to form mRNA. Okay, clear? Now let's move ahead and see further processes. Now we have produced RNA. We have produced RNA. Okay, so yes, so we have till now we have covered our transcription process, how it is formed, how mRNA is formed or hnRNA is formed, and how does it mature. Okay, so that thing we have covered till now. Now we have to cover what? Now we have to cover translation process. And so if we have to study translation, then we need to know what are the different types of RNA, because translation of RNA leads to production of proteins. So RNA are of three types: mRNA, which stores genetic information; rRNA, which has a catalytic role which shows a catalytic role in translation; and tRNA, which carries specific amino acid with respect to particular codon present in mRNA. Okay, so it forms a machinery like this. So this machinery is mainly comprised of rRNA, in which there is a T-shaped or a plus-shaped tRNA, which carries specific amino acid here, and it reads, the tRNA reads information present on this mRNA, the codon, it reads codon because it has anticodon, and it adds specific amino acid with respect to that codon in the protein chain, growing protein chain, and that's how this machinery keeps on moving ahead and ahead. Okay, and hence it forms a long chain of amino acid, and at the end, when they get near the stop codon, or they get on the stop codon, which are UAA, UGA and UAG, then release factor binds on this machinery and this whole complex breaks down, because stop codon, they don't have amino acid. In nature, there are no tRNA which have amino acid specific to these three stop codons. Okay, that's why release factor bind and this whole machinery gets disintegrated. So the tRNA, the rRNA and the mRNA, they gets disintegrated, and the protein chain is released in the system, which starts to modify itself with the help of different machinery like chaperones, and after modification it becomes active for its structural and functional role, and hence it actually controls the what? It actually controls the different traits of our body. Tick. Now, these genes are not always expressed in our body. है ना? These genes are expressed when it is needed. So it means that the gene expression is highly regulated, and it is studied. It was studied first by Jacob and Monod in case of E. coli, and they basically studied a lac operon, an operon system which is known as lac operon. It's a catabolic system which is involved in giving energy to the cell. Or E. coli, it actually breaks down lactose to produce glucose and galactose, and hence these glucose are then further used in respiration to produce energy. Tick. So we know that the primary source of energy is glucose, but lactose is also used as an energy source when glucose is less. So this operon, which is known as lac operon, it is involved in breakdown of lactose when glucose is less and lactose is high. This operon has certain set of genes like the promoter of inhibitor gene, and these ZYA genes. This Z gene codes for beta-galactosidase, this Y gene codes for permease, and this A gene codes for trans-acetylase, out of which this beta-galactosidase gene, it is involved in the breakdown of lactose into glucose and galactose. Then this glucose will undergo respiratory process. Okay, but this gene is only activated once the level of glucose goes down, because if glucose is present, then why do we need to break down lactose? है ना? And how it is switched off? It is switched off in normal condition because this inhibitor gene, it produces a protein known as inhibitor protein or repressor protein, which binds to the operator site of this operon, and it do not let the RNA polymerase to polymerize or to transcribe this Z, Y and A gene. But if glucose is present in less amount and lactose is present in high amount, then lactose binds with this repressor and changes the conformation of this repressor, and the change in the conformation of repressor actually leads to what? To decrease in the affinity of this inhibitor towards this operator region. That's why this inhibitor or repressor do not bind to the operator region once lactose is bound to it. Okay, and hence in the absence of glucose, this thing is switched on and this beta galactosidase is produced, which breaks down lactose into glucose and galactose. Okay, so that's how gene regulation is done. This is a very simple case, but it is also done in any kind of gene. Okay, but one thing which is very important is that this inhibitor is a constitutive protein. It is a constitutive protein because it is produced anytime or every time in the cell of E. coli. Now, one thing that is important for you, which is remaining in this chapter, is to know about DNA fingerprinting. Okay, now our DNA, its 1% is coding region and 99% of our DNA is non-coding. Okay, but among this 99% DNA, there is some part of DNA which is polymorphic. What do you mean by polymorphic DNA? That polymorphic DNA has same type of sequence repeating multiple times, like ATGC, 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 ATGC repeating 100 times. That type of non-coding DNA is known as polymorphic DNA. Polymorphic DNA can be of many types, like single nucleotide polymorphism or satellite DNA. Satellite DNA are of two types, mini satellite and microsatellite. Mini satellites are used in DNA fingerprinting process. And this mini satellite, it has VNTR regions or variable number of tandem repeats in them, which are unique or which are universal for particular individual. So these VNTRs are different for different individuals except monozygotic twins. And these VNTRs are compared in different individuals to compare their DNA fingerprints. And that's how the universality or specificity of particular individual is noted or is recognized using DNA fingerprinting. Like if I give you one example of a crime scene, let's say from a crime scene we got a blood and we extracted the mini satellites whose bending pattern looks like this. The VNTR showed like this. Okay, and we suspected that A, B, C, D. Among these A, B, C, D, one of them is the criminal. And the person who is criminal, his bands will coincide with these all bands. And if you note down or if you see, try to watch all the bands here, which one among these, whose bands among these are coinciding with these bands? It's C, है ना? So the person C has all the bands coinciding with these whole bands. So that's why he is a convict or he is a criminal. And that's how we study DNA fingerprints. Okay, clear? So this is a way how we study DNA fingerprints. Its process is there written in your book that you should do by yourself because it's not a very tough job, but you need to know how to read a DNA fingerprint. Okay, clear? Now, so till now we were studying about molecular basis of inheritance, which actually told us or which actually led us to understand how traits are transferred from one generation to another generation, next generation at a molecular level. Now, we will study mainly those chapters which are actually studied as a macrobiological chapters. Okay? So evolution and ecology are two chapters which are done at a macro level, but yes, there are some other chapters also that will cover, like biotechnology is also there. Okay, let's see. So in evolution chapter, we will be mainly covering 2-3 things. First one is Darwin's theory of natural selection, second one is homology, analogy, adaptive radiation, and third one is Hardy-Weinberg principle. Let's study. So Darwin, he gave his theory of natural selection based on his findings in the Galapagos Islands and the world tour that he did on HMS Beagle ship, है ना? And he gave his 5 points, which are OSVSN, which is that whenever there is overpopulation, then there is a scarcity of resource or resource limitation, which leads to struggle for existence, and this struggle leads to competition. And due to competition, there are variants which are present in nature, and some of the variants which are better able to survive that stress are selected, naturally selected, and other ones, they themselves get deselected. But deselected term is not so good to use, but yes, the better ones get selected. So survival of the fittest, whenever we say survival of the fittest, the term fittest actually do not mean to say that it is physically strong. It actually tries to say us that the person is reproductively fit or reproductively active or is able to lay the progenies. Okay, this is Darwin's theory of natural selection. Now there is one very important term which is known as adaptive radiation, which was actually which was seen by Darwin in his tour to Galapagos Island in the Darwin finches, and also it is seen in Australian marsupials also. So if we talk about adaptive radiation, what does it mean? That if we have a common ancestor, but these ancestors, they start to live in different ecological niches, then due to different type of environment, due to different type of ecological conditions, they evolve different type of trait, and they become different. They start to show variations, and that divergence or that radiation is known as adaptive radiation, and it is mainly seen, or it is seen in many individuals, but the two examples that is important for you with respect to exam is adaptive radiation in Australian marsupials and Darwin finches. So Darwin finches showed variation in terms of their beak shape. Some had very pointed beak, some had broad beak, some has average sized beak, so such things. Okay. Now homology and analogy. What do you mean by homology and analogy? Analogy and homology, they actually try to tell us whether the ancestry, whether we actually try to compare two individuals or many individuals, more than one individual, and this comparison leads us to understand whether these two individuals has a common ancestry or they have a different ancestry. If they have a common ancestry, then they are going to show similar body structure. Anatomical structures of their body will be same, but the function performed by those structures, body structures, may be different. Okay? But more or less, homology denotes common ancestry and divergent evolution. Adaptive radiation is also an example of homology. Okay? Now analogy denotes different ancestry. Analogy denotes different ancestry, and due to different ancestry, they have different body anatomical structures. Okay? But due to their living in same ecological niches, they start to perform similar function. And this analogy is an example of convergent evolution. Like the wings of bats and wings of birds, they are both used for flight taking, but they looks very different. Their anatomical structure are very different because bird is ape and bat is a mammal. Different ancestry, but same function. Okay. Now, Hardy-Weinberg principle. Hardy-Weinberg principle, what is it? Hardy-Weinberg principle says that if a population is not evolving, then the allele frequencies, capital T, small t, allele frequency P and Q, their sum will be equal to 1. if a population is not evolving, or if the gene pool of a population is constant, then their allele frequency, then the sum of their allele frequency will be equal to 1. P plus Q equal to 1. And also their genotypic frequencies, P square plus Q square plus 2PQ will also sum up to be equal to 1. Okay, but the point here is that how does a population not evolve, है ना? We know that populations, they tend to evolve. Yes, this is a true fact that population they tend to evolve, and they always evolve. But the extent of their evolution is different. ठीक है? But this principle, Hardy-Weinberg principle, only applies if and only if population is not evolving. So generally, whenever we do a research, genetic research, we do not get P plus Q is equal to 1 or P square plus Q square plus 2PQ is equal to 1. It always come out to be 0.8, 0.7 or 1.2, 1.3, because never Hardy-Weinberg principle is fully applied or fully correct. So the population, they evolve due to these all characteristics: migration, natural selection, genetic drift, recombination and mutation. These are the 5 characteristics which leads to evolution of a population, or which leads to actually change in the gene pool of a population. But if these all things are not happening in a population, then only the population will not evolve at all. But we know that always in any population, one among them will be happening. Okay, clear? This is your Hardy-Weinberg principle. Now, next. So next chapter is biotechnology, which is actually dealing with recombinant DNA technology. So in recombinant DNA technology, we are more concerned to produce a protein of our interest outside the body of any individual. Let's say insulin, which is produced in our body, if we are able to produce it in the outside media, then it's very beneficial for us. We can sell that insulin in the market, है ना? Let's see. So recombinant DNA technology, basically what does it tells? It tells that we can express a DNA, a gene outside our body to produce a protein. Okay, how can we express it? We first have to extract our gene of interest from the whole DNA, and that we can do by digesting this whole DNA with an enzyme known as restriction endonuclease, like EcoR1, which cuts at GAA-TTC. So the restriction endonuclease, they cut at specific recognition sites or known as palindromic sequences. Okay? So once we'll cut the whole DNA, we know that this whole process is happening in a test tube. And once we cut our DNA of interest, we know that multiple fragments of DNA will be produced. But we need only our fragment of interest, which is present here. How can we separate our DNA of interest from the other fragments? We can simply do a process known as gel electrophoresis. So that the DNA bands or the different fragments of DNA run in different so the DNA fragments, they run as per their body weight and charge. Okay, so we tend to get different fragments of DNA on the basis of their body weight, on the basis of their molecular weight. The lightest DNA moves the farthest, and the smallest DNA, it remains at the very initial site. So we just need to know the number of bases present in our DNA of interest, and we can cut it and separate it out. Okay? By just illuminating a UV light on the gel that we have prepared. Because the gel that we prepare, we add ethidium bromide to it, which gives bright orange color to the DNA. Once UV light is illuminated on it, and then after cutting the DNA of our interest, we just purify that DNA from the gel using a process known as elution. Okay? Now once we are done extracting our gene of interest, we have to express it outside the human body, let's say. But if we have to express it outside the human body, we have to express it in some other organism, okay, like a bacteria. So we can express it in bacteria, express the gene coding for insulin in a bacteria, but we have to first transfer that gene coding for insulin inside the bacteria, and that is done by using a vector. And vector, most commonly in laboratory, the vector is plasmid. Plasmid is an extracellular, independent and autonomously replicating DNA that is mainly present in bacteria only. Okay? So once we have extracted our gene of interest, then we cut the plasmid of our interest by a restriction endonuclease, and then we ligate our gene of interest in this plasmid, okay, with the help of DNA ligase enzyme. Okay. Now once we have got a plasmid containing our gene of interest, then we simply transform it inside a host, and the host in most of the cases is a bacteria, or else it can be a plant cell or an animal cell. Okay? And it is inserted inside a cell by using different methods like heat shock treatment, like retroviral vector, using retroviral vector. We can use gene gun also in order to transform it into a plant cell. We can also use agrobacterium tumefaciens vectors in order to transform it into a plant, and we can also use micro injections to transform it inside an animal body. Okay, clear? Clear? Now let's move ahead. Now let's move ahead and see what happens next. Once we have inserted our plasmid of interest inside a host, then what will happen is that this plasmid, which is autonomously replicating DNA, it actually starts to express itself, and once it will start to express itself, it will also express our gene of interest. So it will produce a protein of our interest that will purify using downstream processing, and hence we can market it as per our need. Okay, clear? Now there is one very important topic which is remaining in this chapter, is selectable marker. Is what? Is selectable marker, from which questions are asked every year. From which questions are asked every year. Okay, let's see. So we know that we will try to, of course, insert our gene of interest inside a plasmid, है ना? Inside a plasmid. But this reaction happens in a test tube, and we are not able to see that whether it is going to cut by restriction enzyme or not, whether it is ligating, or if the plasmid is restricted by restriction endonuclease, then whether our gene of interest is inserted inside that plasmid or not, है ना? So these all things we can't see it, we just have to add the enzymes and let the reaction to happen. But we also can't assume that if we have done all the things, then all the things would have been done, है ना? We can't assume that thing that it is done, है ना? We have to recheck it, cross-check it by using selectable marker. What are these selectable markers? Selectable marker are basically some genes in the plasmid which tells us that whether our host in which we have inserted our plasmid, that contains a plasmid which has our gene of interest or not. Okay, and it is done with the help of two markers or two genes. One is the antibiotic resistance gene and one is the beta galactosidase genes, which makes us to get sure that whether our gene of interest has been inserted in the plasmid or not. Okay, let's see them one by one. let's see them one by one. So first one is the antibiotic resistance gene. So let's say the plasmid in which we are inserting our gene of interest contains a gene which makes that plasmid resistant to antibiotic ampicillin. Ampicillin resistance gene is there. So if we are able to insert our gene of interest in between the ampicillin resistance gene, then we know that ampicillin resistance will be gone. And if it is not inserted in that ampicillin resistance gene, then the ampicillin resistance will not be gone. And that's how we do it. We first try to insert our gene of interest inside that plasmid, and then we'll try to select it by growing it in the ampicillin containing medium. If it is able to grow in the ampicillin containing medium, then we know that it is not inserted. But if it has been inserted in the ampicillin resistance gene, then it will not, the bacteria or the host containing this plasmid will not be able to grow in the medium containing ampicillin, because the ampicillin resistance gene has been inactivated due to insertion of our gene of interest inside it, and that is known as insertional inactivation. Clear? Clear? Now let's move ahead and see blue-white screening. So this method of antibiotic resistance genes, it needs multiple plating, which makes it a chaos. That's why we use a blue-white selection method as a selectable marker. So we have a plasmid known as PUC8, and this plasmid contains a LAG-Z enzyme that was used in case of apna lag operon. This LAG-Z gene, it codes for an enzyme known as beta-galactosidase enzyme, and this beta-galactosidase, it breaks lactose into glucose and galactose, but it is also specific to another substrate known as X-gol. It also breaks X-gol to produce some, it also catabolizes X-gol to produce some constituents, and it produces a byproduct which gives blue color to the cell in which this plasmid is present. Okay, so if this LAG-Z is active, then that colony will give blue color. But if we are able to insert our gene of interest inside this LAG-Z gene, then that will not give a blue color. That will tend to give a white color, because it will not give a blue color, because LAG-Z gene has been inactivated due to insertion of our gene of interest here. So if it is white, then we have got our transformant. If the colony is blue, then it is a non-transformant colony. Okay. Now, this chapter has one more topic which is very important, and it is PCR. Question was asked in 2024 IIT exam also. And this PCR is basically a method to amplify the gene of interest or to amplify any DNA. Basically, it's a DNA replication step in a synthetic media, which is done by using 3 steps: denaturation, annealing, and extension. In denaturation, we generally give a very high temperature to the DNA, 94 degrees Celsius temperature to the DNA, so that the hydrogen bonds, all the hydrogen bonds in the DNA get broken down, and the double-stranded DNA had become a single-stranded DNA. Okay, like this. Now, in the next step known as annealing, primers are added. Primers and specific buffers, DNTPs and magnesium dipositive ions are given to the medium, so that primers are able to bind to the single strands. Okay, and once they are bound to the single strand, a specific DNA polymerase is added, which polymerizes the whole DNA, by the polymerization steps using these DNTPs as substrates. Okay, and it is done at 72 degrees Celsius. Now at 72 degrees Celsius, normal DNA polymerase will of course get degraded, but this Taq polymerase is a specific DNA polymerase which is active at high temperature because it is extracted from a bacteria known as Thermus aquaticus, which is found at 100 degrees Celsius in the thermal vents. Okay, so this is a basic process of PCR which involves 3 steps: denaturation, annealing and extension at 94 degrees Celsius, 50 to 60 degrees Celsius and 72 degrees Celsius respectively. Okay, clear? Now this is a type of exponential growth because 2 to 4, 4 to 8, 8 to 16, 16 to 32, 32 to 64, such type of things are happening here. So that's why it follows an equation known as n is equal to n naught 2 to the power n, where n is the final DNA amount, n naught is the initial DNA count, and 2 to the power n, there's this small n, it denotes number of cycles or the number of generations. In bacteria it is known as number of generation. In DNA it is said to be as number of cycles of PCR it has undergone. Okay, now the next or the last unit that we have to cover is, of course, what is, of course, ecology. It's a macro study. It's a macro study which studies populations and communities. Okay, so we'll cover the 3 chapters of ecology in a composite manner. We'll cover the important topics of those 3 chapters here in this session. Okay, first thing is that how do you calculate whether a population is growing or it is degrowing? How much is the number of population? It can be simply calculated by noting these points, like number of individuals at time t is equal to number of individuals at time 0 from which we started, plus birth plus immigration increases the count, and death plus emigration decreases the count of population. Okay? This is a simple formula. Now next thing is the population growth curve. A population growth curve can be of 2 types: exponential and logistic. Logistic growth curve is more realistic, while exponential is not so realistic. And it's if we plot a graph between number of individuals with time, then the exponential growth curves is a J-shaped curve. It looks like a J shape, and the logistic growth curve, it looks as if it is an S-shaped curve. Why logistic growth is more realistic? Because in logistic growth, we take into account or we consider that the amount of resources present in our ecosystem are limited, which is a truth statement. But here we assume that the amount of resources in the environment present in the ecosystem are not limited, are unlimited, which is not the true statement. It's a false statement, because the resources are always limited on this earth. This earth itself is a limited thing. Okay, now let's compare them one by one, exponential and logistic growth curve. If we compare, it assumes that resources are unlimited, it assumes that resources are limited. Okay, logistic growth curve, it takes into account carrying capacity, but exponential do not have such assumptions. What is carrying capacity? Carrying capacity is the maximum number of individuals that a population can support. More than that it can't support. Okay? And the formula are typical formula. Exponential growth curve, it will show a formula of n is equal to n naught e to the power rt. There is something exponential. dn upon dt is equal to rn, where dn upon dt is the rate of increase or rate of change of the population, and n is the number of individual at any given time interval t, and r is birth minus death upon time, which means birth rate minus death rate, or else it is known as intrinsic rate of natural increase. Okay, while the logistic growth curve, it shows or it follows a formula dn upon dt is equal to rn k minus n upon k. It looks very similar to exponential growth equation till here, but it adds a ratio of k minus n upon k extra here. Okay? It is multiplied by this ratio also. Take next we need to focus on one thing, that is the age pyramids. We know that a population, let's say human population, we can be divided into 3 parts: children's, reproductively active individuals, and old individuals, or else pre-reproductive age, reproductive age, and post-reproductive age individuals. Okay? If the amount of pre-reproductive age individuals are most followed by reproductive and post-reproductive, then that population is expanding. if the pre-reproductive and reproductive ages are age individuals, the numbers are same, then they are stable, then they are in a stable equilibrium, or the pyramid is stabilizing pyramid. And if the pre-reproductive individuals are less and reproductive are more, then it is known as declining population, because pre-reproductive individuals should be more in number. Out of these three, the best one is the stable type of pyramid. Okay, clear? Let's move ahead. Now, the very important point, the very important topic, subtopic of this chapter is biotic-biotic interaction. We know that whenever we study a population or a community, we know that we are going to study interaction among two individuals of same species or different species. If the individuals interact in a positive manner or negative manner, or they are neutral to each other, those type of interactions will be studied here. Okay, so there are mainly, as per your syllabus, there are 6 interaction which are written over here. The first one is known as mutualism, in which both the individuals benefit from each other, and that is known, and that is an obligate type of relationship, because both of them will live with each other and they can't live without each other. They can't exist individually. They will always exist in a pair. Tick. So both of them benefit from each other. Commensalism, in which one organism benefits for the other, but the other do not have any significant advantage or disadvantage from the other one. Okay? Parasitism, it involves in this interaction, one of them benefits while the other is harmed, but that person who is harmed is not killed directly or instantly. But in predation, same type of interaction is happening. One is getting benefited, one is harmed, but the person who is harmed is killed or dies instantly. Okay? In competition, both the competing counterparts are negatively affected, and in amensalism, one is negatively affected but other one is neither positively nor negatively affected. So these are the 6 interactions that we needed to study. The thing that I told here are the basic principles, and if you know these basic principles, you will be able to solve questions that will be asked in the exam. But I urge you to study the different examples that are there written in your NCERT with respect to all these interactions, because this topic is very important. Okay, clear? Like there is a topic of brood parasitism which is mentioned in your book, which occurs between cuckoo and crow. But if you don't know, if you haven't studied that example, then I think you will not be able to mark those questions correctly if such questions will be asked from the example part of this interaction topic. Okay, so you should study the examples of all these interactions from your NCERT. We don't have much time, that's why I'm not trying to cover all those examples. I'm just giving you an overview of what kind of interaction are these ones. Okay, clear? Let's move ahead. Now, in an ecosystem, we know that plants are the one which produces their food by themselves using sunlight. So they fix sunlight or the light energy into chemical energy, which are used by their body and which are used by the other individuals like herbivores and carnivores, directly or indirectly. And that energy which is fixed by plants, they flow in the whole ecosystem. So whatever energy they fix it in the form of biomass, and that productivity is known as primary productivity. The total biomass or the total energy that is fixed is known as gross primary productivity, but some amount of energy is used by plant itself. So all the energy is not laid down in the form of biomass. Some of it is used as per respiratory losses, and the remaining energy is known as net primary productivity. So basically net primary productivity is equal to gross primary productivity minus respiratory loss. Okay, and this net primary productivity is the biomass which is available for the consumption to herbivores, because herbivores eat what? They eat plants. So they will eat what? They will eat a plant biomass, and the biomass which is available to herbivore is actually the net primary productivity, because the rest has been used by plants itself in respiratory processes. Okay, now let's move ahead and see food chain. So in our nature, we have two types of food chain. One is grazing food chain and the second one is detritus food chain. The grazing food chain is the simplest food chain, which includes plants, herbivores, carnivores, top carnivores. Plants are known as primary producers. Herbivores are primary consumers. Secondary consumers are carnivore, and tertiary consumers are top carnivores. Okay. Though we are more able to see such kind of food chain in our terrestrial ecosystem, but this type of food chain is involved in the energy transfer mechanisms more in oceans, not on land. On land, the major conduit of energy transfer is detritus food chain, which starts with decomposers. So it starts with dead and decaying matter, which is decomposed by these decomposers. Okay, clear? Let's move ahead. Now the process of decomposition or detritification, detritivores actually do this process of decomposition, and this decomposition is a five-step process. Which starts with fragmentation of the dead and decaying matter, followed by leaching, in which lots of inorganic substances, they are swept away deep down the soil, which accumulates as an unavailable inorganic substance, which is known as leaching, followed by catabolism, and after that mineralization and humification happens. In mineralization, all the inorganic matters are solubilized in the soil, and in humification, all the organic substances are laid on the outer rough surface of the soil, which is known as humus, which is a dark amorphous substance, and it is water repellent. That's why it is able to hold water on the soil. That's why humus rich soil is very beneficial for agriculture, because it gives time for the absorption of water on the surface of soil, because it is a water repellent. It do not directly lets the water to flow inside the soil. Okay, now ecological pyramids are also very important topic, because question has been asked from this topic in IIT as well as NEST. So these ecological pyramids, they actually are the pyramids which gives us information about what is there in each trophic level: primary consumer, secondary consumer, tertiary consumer, or primary producers, है ना? Now, so we can compare the different trophic levels in terms of number of individuals present in them, or amount of weight or amount of biomass in that ecosystem, or the amount of energy present in that ecosystem, है ना? If we consider about number and biomass pyramid, these pyramids can be upright also. It can look like this, or else they can be inverted also. It can also look like this. Okay, so both of them can be inverted, and one examples of inverted biomass, inverted ecological pyramids has been written here. Ecological pyramids of numbers can be inverted in tree ecosystem, and biomass pyramids can be inverted in pond ecosystem. But energy pyramid is always going to be straight, okay? Because of this 10% law. We know from 10% law that the amount of energy which is present in the lower trophic level, only 10% of that energy is transferred to the next trophic level, and only 10% of that is transferred to the further next trophic level. So that's why the lowest trophic level will have the largest energy, followed by the subsequent upper one, followed by the further upper trophic levels. Okay, clear? Clear? Let's move ahead. Now, the last chapter of ecology is biodiversity, and it has one important topic which is known as different type of biodiversities or different type of diversities. So we have three different types of diversities. One is genetic diversity, second one is species diversity, and third one is ecological diversity. Genetic diversity basically is concerned with the variation among the species. Okay, like we know that we all are humans, but we have different human races. We have Caucasian population, we have Asian population, we have African population, है ना? So we have different variations among one species, that is Homo sapiens. That is known as genetic diversity. One very important point is the presence of a Himalayan plant known as Rauwolfia vomitoria, which is of different types, and it produces different type of chemical counterparts of one particular chemical, that is known as reserpine. Reserpine chemical is used as an anti-cancerous agent, but it has a variant. It has different variants. And how those variants are produced? Because these Rauwolfia also have different types of subspecies or variants in them. Okay, clear? Now, if we compare diversity at the species level, we will get to know that there are multiple species present in our ecosystem. Like if we compare Western Ghats with Eastern Ghats, we will come to know that Western Ghats have far more species diversity of amphibians as compared to Eastern Ghats. Okay? So if we compare the number of species in particular places, then that will come under species diversity. And the last one is ecological diversity, in which we compare ecosystems, the climates and the type of conditions in those different ecosystems. Like if we compare Gulf country with India, we know that Gulf country, they mainly have deserts. They only have desert. So there is no ecological diversity in them, only desert. But if we compare India, India has Himalayas also, mountains also, we have plains also, we have mangroves also, we have tropical rainforest, we have tropical deciduous forest, we have different kinds of area, we have deserts also. That's why in terms of different types of climatic condition, different types of terrestrial and aquatic habitats, India is a more ecologically diverse country. Okay. Now, this biodiversity, it can be studied in two manner. Okay. One is species area relationship, another one is latitudinal gradient. If we talk about latitudinal gradient, we know that most of the sunlight falls at the equator. And we know that the place where sunlight is the most, the sunlight will be absorbed the most by plants. And plants, they will produce more amount of biomass there because there is more sunlight. So equator is the most biodiverse area because of presence of lots of energy and biomass at the equatorial level. But as we start to move towards pole, the amount of biodiversity, it starts to decrease. And this is a trend here. This is a trend that the amount of biodiversity, it keeps on decreases as we start to move from equator to poles. Okay, this is one thing. Now, species area relationship. If we talk about species area relationship, if we plot a graph between the species and the area, it will look like this. Initially, with the increase in area, the amount of species will also increase. But a time will come where you will tend to increase area covered, but the species number will not increase at all. Okay, so such kind of study between species and area is known as species area relationship, and it actually tends to give us this kind of formula, which is S is equal to Ca2 power Z. S stands for species richness, and C is a constant, A is area covered, Z is regression constant or regression coefficient, and regression coefficient also gives us idea of the number of species or the diversity of species at any particular given area. Okay, now We know that we have lots of biodiversity, but that biodiversity, we are losing that biodiversity day by day. And what are the major causes behind this biodiversity loss? There are 4 major causes behind biodiversity loss, and these are mentioned over here. First one is habitat loss and fragmentation. So if we cut down the forest, so habitat of the natural habitat of all the animals and plants are gone. So that's why biodiversity loss is the major cause. Habitat loss and fragmentation is the major cause of biodiversity loss. Okay, or else overexploitation, illegal poaching, illegal killing of animals. It also leads to biodiversity loss. Alien species invasion, like if we introduce any alien species into an indigenous area, then that alien species, it will not have its natural predator here in this new population, because its natural predator was present in some other area from where you have brought that alien species. Now, in the absence of the natural predator, these alien species, they start to grow abnormally high, and they affect or they start to overexploit the indigenous species, and due to which the indigenous species, they get wiped away. This is also one of the major cause of biodiversity loss. And the fourth one is co-extinction. Let's say if 2 individuals or 2 species are able to survive with each other, they can't survive without each other. Okay, 2 individuals can't survive without each other. Both of them will survive only when they are present close to each other, or else they will not survive. Okay. Now, in that case, if one of the counterparts is gone, then the other will also get away. Because why it will get away? It will get away because they were obligate. They were obligate. They can't grow without each other. Okay. So if one is gone, then other will automatically go away. Okay, so these are the main 4 causes behind biodiversity loss. These are also known as Abel quartet. Okay, now the last topic of this chapter is biodiversity conservation. So we always try to conserve biodiversity, though we are not able to conserve. We can see from what is happening all across the world, Amazonian rainforest is also getting wiped away. But yes, we always try to conserve biodiversity at different levels. Communities, there are many communities which try to conserve biodiversity. Their government also, they put many policies to conserve biodiversity. But yes, more or less, it's a kind of we can say it's a kind of we are trying to conserve it, but we are not, because of different reasons. So let's see how we can conserve biodiversity. It can be done in 2 ways: in situ and ex situ. In situ conservation is basically that mode of conservation in which we actually try to conserve the species or the flora and fauna in the places where they are getting wiped away. Like illegal poaching is causing, like if there is some forest in which illegal poaching is causing decrease in the tiger population, then government actually do what? Government actually starts to recognize that area as a national park or a biosphere reserve or a hotspot, biodiversity hotspot, or a wildlife sanctuary. And they start to employ the forest rangers and police and all those officers in that forest to conserve the flora and fauna present over there, and to restrict illegal poaching and hunting of the endangered animals. So in such cases, we are trying to protect animals and plants in the area where they tend to grow, at their site, on site conservation. But here in ex situ conservation, we do not go on the site where illegal poaching is taking place, or in the forest. We don't go in the forest to conserve the flora and fauna which are being wiped out. In ex situ conservation, we actually try to take those flora and fauna from their natural habitat to our gardens of our homes or to the zoological parks or to the botanical gardens, and we grow them there so that they can grow properly in those defined conditions which are not their natural condition, though we try to give them their natural condition, the conditions like their natural conditions. Okay, so such kind of thing is known as ex situ conservation. In ex situ conservation, we also try to conserve the seeds, sperms, semen and spores of different endangered or critically endangered species who have a high risk of extinction. Okay, we conserve them in liquid nitrogen under minus 196 degree Celsius. Okay, so these are some of the methods of conservation of biodiversity, and with this we are ending up with ecology. Okay, hope you have enjoyed this session, and I hope this session will help you a lot in understanding the most important topics of biology, important for your IIT exam. But at the end also I am saying this thing, that the topics that I have covered, they are utmost important, but the topics which we haven't covered or the factual topics, they are also very important, because they can straight away give you 4 marks in just 5 to 10 seconds, because you have to just know the answer and tick mark them. Okay, so you have to cover these points, these chapters also, but you have to cover those chapters also which are factual. You just need to do some things. You don't need to do many things there, है ना? You just have to write all the facts there in your notebook, or if you don't want to write them in the notebook, then also you can just mark them in your book, whatever book you follow, and just try to read them every day. Read those underlined things every day, and after 8 to 9 days or 1 week, you will be able to see the change, that your subconscious has got those facts in your mind. Okay, so you just have to read those facts every day without taking any tension that whether you will be able to remember it or not. Okay, so that things are also important. So with this, I am ending up this session. I hope that this session will help you. Till then, keep reading, and keep reading, and at the end, all that I can say is best of luck, and keep doing your part of hard work. Okay, bye bye.